think we'll go ahead and get started with the webinar. Before we get started, I'll just mention some of the upcoming talks. So at the bottom, the upcoming talks, since we're doing this every 2 weeks, we'll be in 2 weeks. We'll have Stephen Buna giving a talk on low KV imaging in the SEMs, followed by kind of a different discussion than what we've been doing before and just talking about YCMS with some different staff from around campus. And then getting back to the TM world with Hank, with some talking about some more work that's been going on with the TEMs. For today's talk, we have Giovanna Grandetti. She's been here almost a year now, so it's crazy. She started during all this pandemic stuff, so it's been fun getting to know her with that. But with that, she brought a lot of experience with her from the cryo world, sample prep world. I've even been getting her to do some work on the SEMs as well. So it's been fun doing all that kind of work. And today, one thing that she's been doing a lot of work on here is cryo-EM sample prep. So she's going to be giving a talk, trying to understand that a little bit better for everyone.
All right. Thanks, Dan. So thank you for the intro. So as Dan mentioned today, I'll be going over some troubleshooting with cryo-EM sample preparation. Okay, so first things, a little bit about this webinar format. Feel free to ask questions during the talk. You can use either the Q&A or the chat buttons, or there's a raise hand function as well. If you have some in-depth questions and want to stick around after the webinar, feel free to, and we can have a quick chat. And if you're having any technical difficulties, like if you can't hear me or need me to slow down, feel free to just say something in the chat. I'll try my best to pay attention to it. So one other thing that's always fun to mention is CMAS has a lot of high-end instrumentation here. So a lot of fancy microscopes. And the two dedicated cryo-EMs we have are the Glacios and the Krios. And I will say that the Krios here is probably the fanciest microscope that I've ever used in my life. So it's got all the bells and whistles you could ever want. For additional CMAS resources, if you're here, you're already doing the first bullet point and attending a Beyond the Scope webinar. And if something catches your eye on this CMAS instrumentation slide and you would like to learn more, you can schedule a one-on-one consultation with any of the instrument managers with all of these different techniques. We also have some resources for data analysis, and if you're interested in learning more about that, you can contact cmas@osu.edu. So this is my first Beyond the Scope webinar. So as requested, I'm going to give a little bit of background info about me and what I do outside of science. So usually when I'm not doing science, I like to be outside, and that consists mostly of either fishing, gardening, and trying to convince my kids to get outside and enjoy this beautiful yard that we have. So I'm always happy. I'll talk your ear off about fishing. I could talk about that all day. But before I get too far in the weeds, let's get back to the science. So my scientific background, I got my bachelor's degree. I dual majored in biology and chemistry at Walsh University. I went on to get a PhD in chemistry from Virginia Tech. And my grad student work was using confocal microscopy and flow cytometry to look at gene delivery polymers. So a lot of cell biology background. I did a postdoc at the Cleveland Clinic, and there I learned more molecular biology techniques. So things like cloning, protein prep. And after that, I worked as a postdoc at the NIH. And here's where I started getting into electron microscopy. So I started doing FibSem, and here's where I learned cryo-TEM and negative stain TEM. So after that, I worked as a scientist with Leidos Biomedical Research. And here I worked at a multi-user facility and focused mostly on cryo-EM. So screening samples, optimizing sample prep. And here's where I learned a lot of my like tips and tricks on getting people the best cryo samples possible. So after a while of doing that, I wanted to try some teaching. So I got a job as an assistant professor of chemistry at Muskingum University, not too far down the road at New Concord, Ohio. And as Dan mentioned, I joined here almost a year ago. So I joined here in May, and I focus mostly on cryo-EM research here. So for those of you not familiar with the cryo-EM workflow, here it is. So you start off with your sample prep. So your goal is to have as pure a sample as you possibly can get. Once you think you've got everything figured out, then comes the screening process. And this usually takes a few rounds to get all the conditions nailed down just right. So particularly this freezing and viewing it on the glacios. These two things usually take some time, and sometimes you have to end up going back and changing the biochemistry of your protein if you want to get good data collection. But once you find that perfect grid, you can then move on to the cryos, collect some high resolution data, and then hopefully get it processed. But before we get to that point, as I mentioned earlier, your sample absolutely has to be pure. And there's various ways to check for purity, but there's not a lot of ways to check for your structure before you get to the cryo-EM. One good thing to do before you get to cryo-EM is to do negative stain. So negative stain can end up saving you a whole lot of time and can end up cutting down on some rounds of these two steps here. And I'll tell you a quick story about why negative stain is so useful. So I was working on this engineered influenza protein, and it's a trimeric protein. And for the biochemistry, we were using something called a Foldon motif to hold these three arms of a trimer together. We did our chromatography and everything looked just fine. However, when we went to cryo-EM, we couldn't find any of the trimeric protein. So we had two of these influenza trimers. One was a native trimer and one was a chimeric trimer that we engineered. The native trimer looked fine in cryo-EM. We went straight from the SEC column into the cryo microscope. Everything looked good. So we thought to do the same thing with the chimeric trimer. Everything looked good by chromatography, but when we got to the cryo-EM, it just looked awful. So we were expecting these nice trimers, and we ended up with just a bunch of dots. So we went back, did more chromatography, more purification. We did a melt curve analysis and everything looked good, but no matter what we tried, we just couldn't get it to look good in cryo. So then we took a step back and said, okay, let's bring in some negative stain and see what's happening with this protein. And what we found was that the native trimer had a nice compact structure that stayed together very nicely on the cryo-EM grids. But with our engineered trimer, you could see a negative stain that it's kind of splayed out and floppy. So even though both proteins looked great with all of our biochemistry methods, it took negative stain to figure out what was actually happening. So negative stain is a lot faster, and most of the time it's easier than cryo-EM. You can screen a lot of different conditions quickly, so different buffers, different salt concentrations, and you can even use negative stain to obtain low resolution structures, which can help you for later data processing with cryo-EM data, because it gives you a better idea of what shape you're looking for. Also, depending on what exactly your biological question is, sometimes negative stain is enough and you don't even need to go to cryo-EM. So another example of where negative stain can be useful is for a question similar to this. We were studying a protein called NF1, And we wanted to know what regions of the protein are necessary to form this dimer. So we made a lot of different constructs. And by using negative stain, we were able to very quickly find our answer. You can also answer even more basic general sample knowledge questions. For example, we had some proteins that they had to go directly from the SEC column onto a cryo-EM grid and couldn't be stored for any amount of time or they would just fall apart. Other proteins are much more stable. So you can use negative stain to quickly get an idea of how long you can store your sample. You can also figure out, okay, which pH does my protein prefer? Sometimes if you're seeing aggregation, altering the pH of your buffer can help with that. But if it makes your protein unhappy, then it's not a viable way to move forward. So you can also screen a lot of different buffer conditions with negative stain, but one caveat is that it's not compatible with all buffers because the heavy metal can react with the stain in different ways and cause a lot of artifacts like you see here. So now let's get to the cryo-EM. So you've done your purification, you've done your negative stain, you know your ideal sample buffer and storage conditions, and you're ready to see what this looks like in cryo-EM. For cryo-EM, when you freeze your sample, the freezing absolutely has to be fast because you want vitreous ice. So when you get to CMAS and you're ready to plunge freeze your sample, I always like to make sure that there's a layer of solid ethane around the edges and bottom of this copper cup. Liquid ethane is not enough because ethane liquefies at minus 88 degrees Celsius, and that's not cold enough to get vitreous ice. So some people will get liquid ethane and just quickly move on to the Vitrobot. So I would caution against doing that and always make sure that you have this layer of solid before you put your ethane bowl on the Vitrobot. Also, check the top of your ethane. It should be completely clear. If you see any bit of cloudiness or some ice crystals floating around, what I do is dry off a pair of tweezers really well with a hair dryer and then just pluck at those bits of contamination to try to get a perfectly clear window of liquid ethane. Also, when I have my grid boxes in this folder, I try to keep those under liquid as much as possible. They are safe in the vapor phase, but most of your contamination is going to happen at the air-liquid interface of that liquid nitrogen. So I try to keep that air-liquid interface above my grids. So try to work under the liquid as much as you possibly can. So optimizing your freezing and blotting conditions is a trial and error process. So there's not one set of conditions I can tell you that will work well for all samples. Once you freeze your grids and you're on the glaciers, you want to make the most of your screening session. So what I normally do when I'm screening is I have my TEM grid, which is a circle, and I just go north, south, east, and west. So take images at those corners of the grid and then in the middle. Make sure my ice looks good. Make sure that I have a nice even distribution of protein particles and that I can see different orientations of those proteins. I also want to look for low contamination because sometimes these little black squiggles, you can mistake them for your protein, and they also make data processing very difficult. So if I have a lot of these black squiggles, I just don't even bother moving forward with high resolution data collection. You also want to have as thin ice as you can get away with. So you want your ice to be thin enough to get low background and try to get as clear a picture of your protein as you can. So as much contrast as possible. You don't want too thin because then you end up with a lot of these empty holes. So there is a little bit of a balance there. You also need to have enough good areas for data collection. Like I might have 1 or 2 grid squares that look really ideal, but that's not really enough to get me a full data set. So I try to shoot for a minimum of 10 good grid squares, but that's kind of an arbitrary number. Ideally, you want your entire grid to be like good. So cryo-EM sample prep is a lot more nuanced than negative stain sample prep. So for negative stain, it's pretty difficult to mess up on it. You just put your sample on the grid, blot it, rinse it, blot it, and add your stain. With cryo-EM, you have to worry about ice thickness and what's happening at the air-water interface with your protein. So again, ideally, you want nice, even protein distribution, different orientations, and as thin ice as you can get away with. Now here's my hall of shame of what you don't want to see when you're screening. So if you see a lot of these dark black blobs, this is caused by ethane contamination. And then you could see a nice crack in that grid square. So this is caused by poor grid handling, so you need to take more care when you're doing your sample prep. Sometimes you'll see this really thick ice, usually around the edges of the grid bar. So the grid bars tend to heat up faster than the areas in the middle. So this is crystalline ice. So at some point, this became either devitrified or it never vitrified. So you want to try to avoid that by making sure that you're keeping your samples under liquid nitrogen as much as you can, and making sure you have that layer of solid ethane in your Vitribot bowl. If you see that your ice is too thick and you're not getting much contrast in your images, some things you could try are increasing the blot force and increasing the blot time. Sometimes you'll see these hydrophobic patches and almost nothing in the holes. So this is caused by your grid not being hydrophilic enough. So you can increase the glow discharge time. And then these black crystals, sometimes you can even see them form hexagons. This is that contamination at the air-liquid interface of your nitrogen. So if it's a really humid day, water vapor from the air can get into your liquid nitrogen and contaminate your sample that way. Or what I see more often is people will use their tweezers, they'll have something in the liquid nitrogen, take it out of the liquid nitrogen, and then dip it in again really quickly. When your tweezers are under liquid, they get super cold. When you take them out and leave them on the bench, they're cold enough that water vapor from the air is going to condense on your tweezers. So if you dunk them in your liquid nitrogen again without thoroughly drying them off, you're going to see a lot of this type of contamination. So when you're here, you want to get the most out of your grid making session. So you want to give yourself the best chance of success. Some things you can do to minimize contamination are to keep your grids covered as much as possible. So I usually leave the grids in the glow discharger until right before I'm ready to use them. And I always cover my ethane when I'm not actively using it. So by keeping your grids and your ethane covered, you can hopefully minimize some of this type of contamination. And again, anything that touches the grids has to be cold and has to be dry. So thoroughly dry out anything that touches your grid. And when you're putting your tweezers in the liquid nitrogen, getting ready to pick up a grid, I always cool them off to the side and make sure they're super cold and they reach liquid nitrogen temperature before touching your grid. Because when you have metal tweezers and metal grid bars, it's really easy to transfer heat and warm up your sample. So now we get to the actual troubleshooting. So you've made your grids, you've done everything you can to minimize contamination, but you're still seeing some problems with your grids. So one common problem is that you get to the microscope and you don't see any protein in the holes of a quantifoil grid. So some potential solutions, the most straightforward thing to do is to increase your sample concentration. And I know that in some cases, that's not always an easy thing to do. So like if your protein is produced in mammalian cells, for example, that can get pretty expensive and difficult. Another thing you can do is to pick up the grid with your Vitribot tweezers, leave the tweezers horizontally on the bench top, pipette your sample on, and then wait a minute before putting them in the Vitribot. So this is the way we add sample for negative stain, and it seems to work pretty well if you have a sample at low concentration. My usual go-to is to add detergent, and I'll talk a little bit more about what detergents I add. Another thing that we've tested before and had some success with is adding the protein on the grid horizontally like this, blotting it, then putting the tweezers on the Vitrobot and adding more. Sometimes it works, but I caution users that this method does cause a lot of excess background, because when you blot and now you have these proteins that are drying, you end up with a lot of junk kind of all over your grid. You can also functionalize the carbon to make it less likely that protein will stick there. And you can also add a thin layer of carbon or graphene over the holes of a quantifoil grid. So here's one case where detergent ended up working. So we've tried functionalizing the carbon of this grid. So SAM is self-assembled monolayer. So these are normal carbon grids that we sputter coated with gold and then added a layer of PEG onto them to try to force protein in the holes. And that by itself wasn't enough. We ended up adding some DDM and CHS, and we found that as we increased the concentrations of these detergents, it forced more and more protein into the holes. There's another example here of using some graphene oxide flakes. So with this method, you deposit a solution of graphene oxide flakes on the carbon side of the grid, deposit your sample on the back of the grid, and then blot from both sides to try to get your protein in the holes. And you can see here it worked. We actually got too much protein in the hole. It's not perfect though, because you can see this solution is very non-uniform. So you'll have some holes that look okay, but a lot of holes, you have maybe half a flake of graphene or a fraction of a flake. Another common problem that people see is protein aggregation. So potential solutions, the most straightforward thing to do is to try to lower your protein concentration. You can also add detergent. Another thing that people have found some success with is lowering the blot force and blot time. It does lead to thicker ice, but that thicker ice gives more room for the protein to move around in and gives less chance for the protein to bump into one another and become aggregated. So here we had a protein that would commonly aggregate around the edges of the hole. So when we added some Cymol 7, we got a nice distribution across the entire hole. And one user here named Kai also had an issue with aggregation, but he added some Triton X100 and was able to clear his aggregation problem and ended up with some pretty nice structures. Another user here was working on this fibril sample, and you could see without detergent, we have these really thick bundles of fibrils. So she tried two different detergents. One of those was OG and another was DDM. And with just tiny amounts of those, she was able to get rid of these really thick bundles and can see individual fibrils. So another problem that I always hate seeing is a protein complex falling apart. This ends up being pretty difficult to solve. So some things that we've tried are detergents. So there's a belief that these proteins are falling apart at the air-water interface. So if you can form a shield that stops your protein from getting to the air-water interface by using detergent, hopefully you can stop it from falling apart. For membrane proteins, many groups have found success embedding them in nanodiscs. One interesting thing, and I'm not sure why this works, but sometimes it does, is switching from carbon grids to gold grids. So I'll show you an example of that pretty shortly. Some last resort things I use are either adding a small amount of glutaraldehyde in a technique called graphics or using negative stain to hold your sample together. This lowers your resolution, and I'm always very hesitant about moving forward with data processing when doing these things, because you don't know if you're cross-linking or if you're fixing your protein in a conformation that's not native. You can also try different ways to blot. So we do have a manual plunger here at CMAS, and sometimes blotting from behind or blotting from the very edge of the grid can help protein complexes stay together. Usually though, once we run through a bunch of these type things and go through many rounds of screening, we've found it easier to just change some of the biochemistry a little bit and try to engineer our protein to be more stable. So here's one example where we had a membrane protein that was falling apart. So we embedded it in a nanodisk and put it on a quantifoil grid. And even in the nanodisk, we still found a lot of our protein falling apart. But when we combined the nanodisk with an ultrafoil grid, which contains no carbon, it's all gold, we saw that the protein complex stayed together very nicely. Then we had another protein complex where we were just trying everything to get it to stick together, and changing the biochemistry was not an option for this particular one. So we've tried carbon coating quantifoil grids. We've tried blotting from the edge of the grids and back blotting. And what ended up working for this sample was to add a little bit of nano W negative stain just to hold everything together. But again, it's not ideal because it lowers the resolution of the structure you can get. So yet another problem that you may encounter as you're screening your grids is preferred orientation. If your protein is only facing one way, you're not going to be able to get any structural information on the entire protein. So this can also be a very difficult thing to face. And there's no one size fits all solution. So it's very sample dependent. My usual go-to for preferred orientation issues is to add detergent. If that doesn't work, you can try using either quantifoil multi-A or lacy carbon grids. Those contain a variety of different sized holes, and sometimes changes in ice thickness can help force your protein in different orientations. You can also try functionalizing your grids or adding a thin layer of carbon or graphene, and that can sometimes force your protein to anchor in ways that you can see the different orientations. And then another kind of last ditch effort is to tilt your sample and either use single particle tomography, which is shown here with this kind of flexible protein, or a more straightforward approach is to collect single particle analysis with the stage tilted. Both of these methods will also result in a decrease in resolution, because when you tilt your sample, your electron beam has more ice that it needs to go through. So you're effectively making thicker ice. So that's why we usually try to avoid these types of things. So again, Kai had a protein that wasn't behaving ideally on the grid. It would aggregate into these ladder shapes. So he added some OG detergent, and that was enough to break up these ladders and allow the protein to freely rotate. And again, he was able to collect some very nice high resolution data. So as you can see, most of my troubleshooting consists of adding detergents. So some useful detergents to have on hand are listed here. My usual go-tos are OG and DDM. So these are the two I would try first. If nothing else works, then I would break into the Tween 20 and the Cymol 7. When you're adding detergent, you do change the liquid behaviors of your buffer. So you may also have to play around with the blot force and blot time. So there's some optimization for that. So in conclusion, I have never seen something that worked perfectly on the first try, so don't get discouraged. You can expect a few rounds of screening and some trial and error before you have a grid good enough to get into the cryos. Try to do everything you can to get to the cryos faster, and negative stain is a useful way to let you see what's happening to your sample before you get even into cryo-EM. And when in doubt, just add some detergent. So if you have any questions, here's my email address. And again, I'll leave you with some upcoming events to look forward to. So Stephen will be talking about some low KV imaging on the SEM. Then on March 26th, we're planning on having a staff panel and learning about everyone's different backgrounds and why they chose CMAS. And then April 9th, Henk will take us on an exciting journey into the exciting world of TEM. So for more information on these upcoming events, here's a website you can go to see what's coming up and to sign up for them. And lastly, you can connect with us through all of these different social media platforms. Okay, and that is the end. So if you have any questions, I'd be happy to answer those now.